Thank you very much, Troy. Everyone hears me well? So it's my pleasure to be here. As Troy said, he was one of my former PhD students. Not only him, his brother, also Derek, was also my first PhD student. And I also taught their youngest brother, Weston. He was also in my class. And this is how I lost my hair. Thanks to So as Troy said, I do not do anything with oil and gas and energy and so on. I work with computational biology. But before I talk about the work here, I want to highlight something that first, computer scientists are very agnostic about the data and about the domain. As Troy said, Troy and Derek, both of them did their PhD in computational biology, and Natalie as well here, she did her PhD with me in computational biology. None of them is doing biology now. Right? But the thing is that when you do computer science, you get training in modeling, in algorithms, in how to formulate a problem. To me, the hardest thing in computer science and in data science is how do you formulate the problem well. It's not just about the solution. It's about how you formulate it. You need to understand what the input is, what the output is. Now, having said that, it's very important that not only be agnostic, but also care about the domain. Right? I believe if you want to make impact in biology, you have to understand biology. You cannot go there disrespecting it and say biology is just memorization, computer science is the hard stuff. No, biology is very hard and it's very complex. It's very important to understand it. So when Troy called me to give a talk, we were chatting, I never give this type of talk. I usually go and talk about specific project. I don't talk about uncertainty itself, but I talk about the specific project. I will go into the specific project, but I'll try to make the case here. I'm not coming here to tell you about cancer and biology, but trying hopefully to show you that when you understand how the data was generated, what the biology is, that you model uncertainty carefully as well. So let's actually, the simplest thing for me, a model is an abstract representation of a system to explain its features. And I put some of its feature there because usually when we do modeling, we want to ask questions and about specific properties, and we want to get an answer to them. How detailed the model is doesn't make it right or wrong. The most detailed model is not right, and the least detailed one is wrong. The level of detail in the model has to do with the questions that you are asking. Okay, so if the question can be answered with a very simple model, a simplest model should be. Okay, so simplicity in modeling is not a new topic. People are not talking about simplicity now. Actually, from 4,000 years ago, where Aristotle said that nature operates in the shortest way possible. So this is simplicity, right? I mean, you take the simplest explanation. The more famous one of them is this quote from William of Ockham: simpler solutions are more likely to be correct than complex ones. So I don't know how many of you here have heard of Occam's razor. Occam's razor comes from this guy. Occam's razor, or some people call it the Parsimony Principle, which is always go for the simplest possible explanation. So simplicity in modeling has been there for a long time. Simple models, we have to put Steve Jobs somewhere, but so simple models are not easy. Coming up with a simple model is not easy. Actually, this quote is very correct here in the sense that you want to get a simple model that's also correct. You have to understand, and you have to have very clear thinking, and that's not a trivial thing. In fact, I teach at Rice, and one of the biggest challenges with students is that their thinking about problems is not very clear. They mix up so many things that they cannot even formulate the problem sometimes. Now, about simplicity, as we know, this is a very famous quote from Einstein that everything should be made as simple as possible, but not simpler. And I will show that this is not just a cliche. In fact, when I talk about the problem I'm working on, that we try actually to go from simple and start adding complexity as we need it, not start from the most complex model. Okay? So again, the title for this series is about taming uncertainty and modeling the unknown. I would say the simplest models rarely work in biology because of uncertainty in the data we collect and the unknowns. Right? There is a famous quote from Don Rumsfeld about unknown unknowns and known unknowns. It's actually correct. People laughed at him when he said it. I don't know if he meant it, but the point is that there are known unknowns and there are unknown unknowns, and there are a lot of them in biology. Complexity should be added to the model on an as needed basis. I really think that when we should start with the simplest model, ask what's wrong with it, why it's not answering the question, and start making it more complex, rather than start from the most complex model. So moving beyond these quotes and getting to the actual question I'm looking at now with MD Anderson is to try to model cancer growth from single-cell DNA. There is a very important thing here. Every word here has a special meaning, especially single-cell DNA data. This is actually about a new type of data that biologists can have access to. It's called single-cell data. So when you hear about people are sequencing genomes, the way usually they sequence genomes from humans is that you take a bunch of cells. You cannot just take one cell. You take a bunch of cells, you sequence them, and you get an average of that. So it could be millions of cells that you have access to. Now the field has moved into the resolution of single cells. You can go to MD Anderson. Hopefully you don't go to MD Anderson, but a cancer patient shows up to MD Anderson. Now they are sequencing single cells from that patient. Okay? And we want to try to understand cancer from that type of data because this is the most promising data. Where does complexity comes? I don't think there is any field out there that has more complexity than biology, and it comes because of the variability of biology in both space and time. So on the right-hand side there, I'm showing you the time scale. If you think about it at the highest level or the largest possible scale, if you look at evolution of life on Earth, the theory puts it about 4 to 5 billion years ago that life started forming on Earth. But you also look at events that are happening now as you are sitting here, protein binding is happening in your cells. And these are events that happen at the microsecond level. But these events that are happening at the microsecond level, these are very complicated circuitries. In fact, both Derek and Troy in their PhDs were looking at these kind of networks inside the cell that govern how genes get regulated and so on. These are things that are happening at the microsecond level. But these circuits have been shaped by evolution for billions of years as well. And at the same time, from a spatial point of view, we are dealing with varying complexity that comes due to space, right? I mean, we look at the humans, in meters. You can think about a human being 1.6 meters or something like that. And you can look at mouse. You can go all the way, C. elegans, Drosophila melanogaster. This is the fly. Then you can go to bacteria. You start looking at very different scales. So all this gives rise to all the complexity in biology. And most of it is unknown. Right? Most of it is unknown. What we observe today is just the tips of what happened over 4 billion years. Right? So evolution working for 4 billion years, and we are just having a snapshot today of that. But going back to cancer, so what is cancer? Cancer is a somatic mutation. So what is somatic mutation? Your cells, most of your cells that you see, your skin cells, lung cells, pancreas cells, and all of that, these are somatic cells. What's not somatic cells are germline cells, the egg and the sperm. So cancer usually happens. For example, people develop breast cancer or lung cancer or pancreatic cancer. These are happening in these types of cells. And what really happens is, a healthy individual all of a sudden has a mutation. A mutation, some letter or something happened to the genome. Why does it happen? It's just a mistake. Because cells divide. In fact, I believe it's within a few weeks, none of the cells that are in your body now will be there. Okay, so I think philosophers can be busy with that kind of questions for a long time. Who are you in 2 weeks from now, right? When all your cells are different. But we are not philosophers. The thing is that these cells divide. They divide all the time, especially like skin cells. They divide all the time. And when they divide, the DNA replicates, right? So there is a mechanism inside the cell that's going to create a second copy of the DNA. When you copy a text, if you have a text and copy it, you are going to have typos. The same thing with the cells. It creates typos. These are mutations. Some of these mutations, actually most of these mutations don't do anything. They are not harmful. Every now and then, a mutation will hit a gene that's very important, and that gene leads to out-of-control growth. And that's what cancer is, right? Cells start growing uncontrollably because cells are supposed to die. That's the normal behavior of a cell. There is a mechanism for it to die after some point. And in cancer, they don't die. They start actually proliferating. So this is what cancer is at a very simplistic level. Now, cancer is very complicated. when you look at the cells inside a cancer patient, they are not homogeneous in any way. You don't take a biopsy from a cancer patient, you look at all the cells there, they all have the same mutation. What happens is that we have populations of cells, these are all the cells in my skin. At some point in time, one cell has a mutation. Because of selection, right, that cell for some reason becomes very fit against dying. That cell starts expanding, so it starts replicating and starts dominating. Some point later, another cell mutates, So what you are seeing here in colors are different types of genetic signatures in cells in a cancer patient. So this is now a big thing in cancer biology and in trying to treat cancer is, when you look at a cancer patient, how heterogeneous is the cancer in that patient, okay? Now, why do we care about these things? Why do we care about understanding this mutation and so on? Because, as Troy mentioned, that the field is moving towards this targeted therapeutics. Cancer, I'm sure everyone here knows that cancer has been treated mostly with chemotherapy. Chemotherapy is just poison. You poison the body, kill cells, and hope for the best, that you are killing the cancer cells as well. But of course, it has very bad effects, side effects, and the second thing, it doesn't work in most cases. So where the field is moving is to develop these targeted drugs. What is a targeted drug? Instead of just poisoning the body, no, let's actually understand what mutations happened, in what genes, what happened to these genes, and let's develop drugs that will actually reverse the actions of that mutation. This is not science fiction. There are drugs out there that are based on these kind of discoveries. This is a very famous case of this genomic medicine. This is what's known as the CML. It's a type of leukemia. In this type of leukemia, we know exactly what happens in that patient in the genome. There are two chromosomes, chromosome 9 and chromosome 22. So in every cell in your body, the genome is organized in chromosomes. These are normal chromosomes, so chromosome 9 and chromosome 22. Chromosome 22 has a gene called BCR. Chromosome 9 has a gene called ABL. Biologists give genes names. Now, what happens in the patients that have this type of leukemia is that this part of this chromosome and part of this chromosome get fused, and they create this BCR-ABL gene. That's what happened in these leukemia patients. The pharmaceutical company Novartis, in fact, developed a targeted drug called imatinib or Gleevec, which is given to patients, actually, cancer patients, that targets this specific gene, because the gene is going to form a protein in the cell. So now you can think about something that's going to come and bind to it and stop it from doing crazy things. So this is not science fiction. This is where the field is moving, not chemotherapy and everyone is a cancer patient. Not everyone is a cancer patient. Every two cancer patients have very different genomic signatures, and they react differently to drugs. The situation gets very bad with cancer, unfortunately. So even with this drug, another mutation hits that genome, and the patient becomes resistant to that drug. This is one of the problems always with drugs. We hear about resistance all the time in the area of antibiotics, but they happen in cancer as well. So I hope now you understand why we care about this, finding the mutations and so on, because this is where the pharmaceutical companies want to move to understand specifics for patients. So questions of interest here, what are the mutations that are present in the tumor sample? A patient shows up through the door at MD Anderson. We want to understand what are the mutations in that patient. In what order did they happen? Because as I showed with the heterogeneity picture, the order in which they happen matters, and especially with the issue of resistance. Okay? Because resistance results from a mutation hitting after a drug, for example, has been applied. And what are the subpopulations in this tumor, right? We want to know, is it 4 or 5 or 17? Because they have been implicated in not only resistance to drugs, but also in metastasis. Okay? So in some cancer patients, okay, cancer is in the liver. In some cancer patients, after a few weeks or a few months or a few years, it also has spread into the bones and the brain and so on. What makes one cancer metastasize versus another cancer that stays located in the same organ? So these are the type of questions we are interested in. And as I said in the title there, they drive the modeling. If we are going after these questions, we need a model that allows us to answer these questions. So this is the time to look at these questions because the data is now are being available. This is a figure that shows the drop in the cost per genome. So now sequencing a genome is so cheap. In fact, this is why, I believe at MD Anderson, their goal is to sequence every patient who goes through MD Anderson for treatment, because now it's so cheap to get a genome. In fact, for bacteria, because they are so small, you can sequence thousands of them in a day. For humans, it's still a bit more complicated, but even for humans, the cost of sequencing a genome is very cheap now. So the amount of data is enormous, and the possibility is there for us to sequence genomes and answer these questions. Now, the issue of uncertainty and the simplest model and moving beyond the simplest models. Now, if we have the ideal data, I want to make the argument that the simplest model would solve the questions for us. What is the ideal data? The ideal data, someone says, we need the genomes from different cells in the patient. Okay, we understand the genome is a sequence of letters A, C, T, and G. The sequence is of length 3 billion. Every genome is about 3 billion letters. Okay, so an ideal data set is that we have, suppose you have 1,000 cells, you have 1,000 files on your computer, each one of them has one string of length 3 billion. This would be perfect data, and every letter A, C, T, G, everything is clean and nice. And how do we now find the mutations? It's very simple. Because we know the genome of healthy cell, I can take now a healthy cell, they sequence it and say, here's the genome of a healthy cell. Here's the genome from a cell in a tumor sample. I can look at a position here and its counterpart in the tumor cell. So, well, here it was CG, now it moved to TA. It's a mutation. Right? It's really that simple. If we have the perfect data, the simple algorithm where you just take two strings and compare them, this will actually tell us about all the mutations. If we want to find the grouping of these into clusters, as Derek is going to talk about k-means, but there is a wide array of clustering methods, you can do that. And the problem is really solved. In fact, even if we want to get the tree structure of how the cells evolved, there's an algorithm from 30 years ago, developed by computer scientists, a very famous computer scientist in the area of computational biology, Dan Gusfield at UC Davis. It's linear time, beautiful. You give it 3 billion letters, it will give you the tree in a matter of minutes on large data sets. But the reality is very far from this. And this is where the simplest model is just saying, let's look at them, and every position they differ at, it's a mutation, and we move on. It will be very misleading. And why is that? I will talk about some sources of uncertainty. The first one is noisy data and noisy assembled genomes. So how is the data collected by biologists? And this is where I said, yes, we are agnostic about the domain, but we cannot be agnostic or ignorant of where the data comes from. This is the protocol that biologists use to collect single-cell sequencing data. So how do they do it? They have a biopsy from a patient. They do something known as cell isolation. So they start isolating individual cells from that. They take these and they extract DNA from it. So there are sequencing technologies that will extract DNA. The problem with extracting DNA from one cell, the amount of data you get is so small that you cannot do much inference from it. You cannot actually even get the genome itself. So what they do, they follow that with something known as whole genome amplification. It's a process where you basically amplify the amount of data. So you have this small amount of data in brown. You actually amplify it now. There is a biochemical process that happens. You amplify it so that you get more signal in your data. Okay? Once they have that, now we are not done. You don't get a genome. There is no technology that will give you, here's a sequence of 3 billion letters. It will give you these short, what we call short reads. Your genome will come back from the sequencing technology as a huge file, text file, where your genome has been chopped off into strings or sequences of length 100 to 500. That 3 billion thing is going to come back in segments of 500 letters. These 500 letters, you have no idea where they came from in the genome. There's no restriction that they are not overlapping. This is how they come. Actually, you will get, so each one of these lines here, the orange lines, this is really a small segment coming from the genome. They overlap, and they get, so some parts even of the genome were not covered by this technology. So this is the process that they follow until they get a genome. We have something known as genome. This is a genome that has been assembled. We assume that this is for healthy cells. This process is going to introduce a lot of error. The first one is error in the raw data. This is the raw data here. This is the raw data. What happens is that when you do the DNA extraction, you're going to get error here. When you do amplification, you're going to get more error. In fact, in single-cell data now, when you look at a file of input file of data, the error rate is about 20%. Okay, so imagine I'm giving you an English text. I say, here's a book to read. 20% of the letters in that book are wrong. This is what you get in that one, right? So now you want to actually try to infer something about cancer and mutations from something that has a lot of what seems to be like mutations, but these are just errors, right? And the second thing is the error in the assembled genomes. So even when you have these, and even if you assume these are correct, now you have a very hard computational task. Again, imagine that you have an English text, some book, some Shakespeare play, and I take that Shakespeare play and I split it into small sequences or strings or sentences, and these are going to be overlapping. Okay, so I can take the first sentence to be the first 5 words, the second one is going to be the words 3, 4, 5, 6, 7, and so on, so overlapping. And then I will introduce error to them. And now I give them to you, and I say, can you please give me back the Shakespeare play? Okay, the text of that. This is really what's happening here. This is a very hard problem. It's known as the read mapping problem, where you are taking these small segments of the genome that the technology gave you. You're trying to build the whole genome from it. Now, why do we have errors? Again, this is where we have to understand the sources of errors. I'm not going to go into the details here, but this is the part where a computer scientist is talking to a biologist or an experimentalist at MD Anderson and say, why are errors happening? And this is the data, this is the information they give us, right? This is not something a computer scientist knows about, allelic dropout or missing data or whatever. So the experimentalist told us that sometimes, we have 2 copies of DNA. Sometimes the technology gives us 1. In fact, every cell in your body has 2 copies of the genome. For whatever reason, that technology can give you just 1 sometimes. Another time is that you have, in your genome, you have a mutation. 2 copies have A and B. That part is completely missing in the data that you get back. These are all artifacts of the technology. Another one is this false positive error. The position in your genome has no mutation in it. But because of error in the technology, this is what's known as the false SNV, or single nucleotide variant. This starts happening. So these are the types of errors that you have to understand in the data so that you quantify this uncertainty. Okay. The other is, it gets more complex in biology, that these errors are not uniform. Not all of them happen the same thing. And when the technology makes a mistake, there was a C and it made it a T by mistake. These don't happen uniformly. For whatever reason, I believe it's for the biochemical reaction reason, is that there's a bias that most of the mistakes replace C by T or G by A, right? Not G by T or G by C or anything. No, it's actually most of the mistakes will take C to T, G to A. The reason I'm highlighting this is, again, not to turn you into sequencing experts or anything like that, but to me, this is the important part when you are trying to model uncertainty. You could throw crazy, math at the problem, but this is easier. It's easier to talk to the experimentalists, understand what's happening, and now we can start modeling it. And the other thing is the short mapping errors. These are algorithmic errors. The algorithm took input, will give you an output. This is a study that's not by us, it's by a different group. That basically they took the 5 most popular tools for doing this assembly of the genome. They will take the input, this file of these short reads, give you as output, here's the genome assembled. These are the 5 most commonly used tools on the columns. On the axis here, we have 5 rows that they simulated data that resembles 5 different organisms. E. coli is a bacterium, and S. cerevisiae here is yeast. The reason you modify this is that because different organisms in biology present different complexities. What you're seeing here, on the x-axis in every panel, this is the number of correctly mapped reads. How many of these small segments were mapped correctly to the right location? On the y-axis here, how many of them were mapped incorrectly? So as you see here, there is this curve that's for the sensitivity, where they basically took the correctly mapped reads divided by the incorrectly mapped reads. And these methods come with a threshold. Okay, so you have a threshold, say, I'm willing to accept this percentage of correctly mapped reads. As this threshold shows you, you want a very low number of incorrectly mapped reads, you have to have very small number of correctly mapped reads as well. The more of the map reads that you want to be correctly mapped, the error rate starts going up as well. I mean, these are the most famous methods that are used, especially bow tie there and Stampy at the end. But this shows you that even if the data was correct, the methods are going to give you a lot of wrong alignments of these or map them into the wrong places. Okay? So all these kind of things that we need to account for. This is, I'm showing you an example from an input file that when you get DNA sequences, this is what you get. It will tell you, these tools will tell you, here's the DNA sequence. For every position, look at every letter here, G, T, or A, or C. Under it, there is a letter. These letters are codes for base quality. So every letter in the genome is going to come with some base quality. It says the quality of this, is X, or the quality of this is Y. The higher the quality, the more you trust it. The lower the quality, when you are making inference, if a position here is a G, and the quality of that is so low, even if it doesn't look like the position in the healthy cell, you don't want to call it a mutation. You want to ignore this and say maybe this is error, right? So it comes with this, even when the algorithm mapped this sequence to the human genome or to the reference genome, also comes with another quality score to tell you how accurate that was. So the data really comes with all sorts of information about certainty or uncertainty at the level of the individual nucleotide in the genome, but also on the entire thing, how it is being assembled. This will be an example of, when you have tons of these segments, they will be mapped against this, what we call reference genome. And it will show you, for example, for this site here, the healthy cell has A, but these cells have a C in them, and so on. So this will be like example visualizing input file there. So what does this all cause? Where does this take us? Where that takes us is that at the end of the day, for us as computer scientists, we need to write an algorithm that takes something as input, gives something as output. The input in this case should be a matrix that, for example, on the columns here are the individual cells. So if the biologist sequenced 1,000 cells, we need 1,000 columns. If there are 500,000 mutations, we want 500,000 rows. Zeros and ones. One means that there is a mutation at the position 1 in cell 1. 0 means there is no mutations. Position 3 in cell 3 does not mutate. Okay? This would be the ideal data. This data is there. Biology is like that. But what we see, the observed data you are dealing with is this. Okay? So the observed data is like this. First is that, these red things in red, these are errors. We know that they are there. Of course, we don't know which one they are. Then there are these dashes. And in addition to the error, missing data. You are not going to have a good read of everything in the cell. So this is the truth, which we don't know. This is what we observe. And this is where we want to do the inference from. And again, that's where, think about this matrix. I'm talking about 20% for every column here. 20% of the entries in every column could be wrong. Okay? This is where we start now going from the simplest models to accounting for uncertainty. What this formula is saying, we don't need to really read it. If you look at the G and D, G is the truth, which we don't know. D is what we observe. What we are putting here is just something very simple. It basically says, what is the probability that we observe a 1, given that the truth was 1? Or that we observe a 1, the truth was 0? Or we observe 0, the truth was 1, or the truth was 0. Where do we come up with these parameters? This is, again, based on the knowledge of the technology that's being used. The experimentalist told us that you can basically, there are 2 types of errors. They are not the same. We cannot use one parameter, so you just go with this, right? So now this is a simple probabilistic model to say what I'm observing is not necessarily the truth, and I need to account for it in a probabilistic way. I want to just say a word here about when I came to Rice. In fact, all my PhD and the first, I would say, 7-8 years at Rice, I was not doing anything statistical. All my work was what we call combinatorial. And the reason I moved into statistical domain is this. You cannot account for this without putting a statistical model on it, right? So for me, I moved into a statistical realm not because it's fashion or it looks nice or it's sexier than combinatorial methods. It was necessary. And if you want to account for uncertainty, I don't know how you can account for it without statistical models. So we did this actually just to account for error. The second source of uncertainty is, suppose the data is perfect. The second source of uncertainty is, when a biologist gets these 1,000 cells, these are cells that they just took today. But these cells have been dividing, the DNA has been replicating for days and weeks. We have no access to that. We have no access to that. We are just seeing the final outcome today of that. So as a computer scientist, I went to the experiment. I said, why don't you sequence this from a cancer patient every day? And he said, this is a very invasive procedure. You don't want to do it, right? So computer scientists, we don't think about these things. But to do this from liver, you are sticking a needle that this long actually into the body to get sample from the liver. I don't think anyone is interested in having that done on them every day, right? So that's not something we are going to get access to that data. We have to account for it. So this is not error in the data. This is uncertainty in the evolutionary process that's happening. It's stochastic. DNA is being replicated. Mistakes are happening, not happening in a deterministic way. So the simplest assumption for this is known as the infinite sites assumption. It has been adopted by biologists for a long time. It says something very simple. Your genome is 3 billion letters. The assumption says that no position in your genome during its evolutionary time, as the cells are dividing, no positions will mutate more than once. It's a simple assumption, right? So if you have one mutation, for example, the 5th position mutates. Another mutation, it's not going to happen at position 5, maybe position 1500 or something like that. Why is that assumption, why does it make sense? Because the genome is so big. These are random mistakes. What are the odds that a mistake is going to happen at the same position twice? That's basically the basis for it. It makes sense. Good basis, the simplest model. However, lots of studies in the literature have showed that this assumption, in practice, when you look at data, is violated. Okay? I'm not going to go through these because this is really biology now, but these are information from papers, not our papers, but these are from papers published in the literature. These are some more papers that we highlight here, some of the statements about cases, data sets, different types of cancer where this assumption is violated. So now when this assumption is violated, why is it? What's happening? So here I'm showing just 2 cases. So this cell, we have another copy of it. This cell divided, and the DNA got replicated. So now we have 2 copies of the cell. Another round of duplication here, or cell replication. If you look at this red star here, what happened is that this red star, a mutation happened at this position, and a mutation happened at the same position at some other place. This is what's known as convergent evolution, and this is not infinite sites assumption. So this is just an illustration of, no, that simplest model of mutation that a site is not hit more than once by a mutation doesn't hold. Okay? And especially in something like cancer where the rate of mutations is very high. So again, just that simple thing that either we go from 0 to 1 or 1 to 0 once in the lifetime of a cell doesn't hold. Again, we go to a statistical model here. And this is a standard technique for modeling evolution or change over time. So if I want to say, what's the probability that an A changes to a T over 5 weeks? The technique for that, the simplest techniques, is to use something known as continuous time Markov chains. Continuous time Markov chains is going to give you the probability that you go from X to Y after a certain amount of time. The only thing you need to do for that, the math is established for that. The only thing you need to come up with is something known as the rate matrix. The rate matrix tells us what's the rate of change from 0 dash to 0 dash or 0 dash to 0, 0. For example, if you look here, 0, 0 to 1, 1, we say not applicable. We are saying here it's impossible at one time point that the 2 copies in the genome changed from 0 to 1 at the same time. At the same position, at the same time, in the same genome. We say that's very unlikely. We basically say this. So this matrix, the way we develop it, it's based on our knowledge, our assumptions, and talking to biologists. The math behind it has been established for decades now. It's just Markov chains. So this matrix accounts for this uncertainty in the evolutionary process. The matrix I showed before, or the probabilities I showed before, account for the uncertainty because of the error in the data. Now that we are equipped with these, we need to go back to our problem. So now I have the input that I know has been evolving and so on. With the models of error and evolution, I need to get to the output. What is the output? I need to know the clonal clusters. It's really a clustering problem. I need to know that in this patient, of these 1,000 cells, they cluster into 5 groups. Each one of them has its signature, genomic signature. This is what clonal genotype says. It's just that this cluster has some, so I think when Derek shows the clustering is that when you do clustering, for example, with k-means, there's notion of centroid of and so on. Think about this is that the DNA centroid. For that cluster, this is the DNA sequence that represents it. And the clonal phylogeny, how these clusters have been evolving, right? So these are not just clusters that sit there. They actually have a tree structure that connects them. So it's a very much more complicated problem than standard clustering. So for this, we went to this more complex model, and the method is known as cyclone fit, that where we put the data, we will put these models of error and so on to capture all of that. This is really the model. This is all there is to it, and I will try to wrap up very quickly. I don't know when I started, actually. So what this model here is what we call a plate diagram for a probabilistic model that will capture the data. What you see here is that D there, this is the observed data. This is that matrix of zeros and ones that we got from the sequencing technology after we did that. This is governed by some clustering. There is a clustering, some clustering mechanism that says these are coming from this cluster, these coming from this cluster. So CJ is a cluster indicator. So C1 is 5, which basically means cell 1 came from cluster number 5. C2 is 17, saying cell 2 came from cluster 17 and so on. This one here is the clonal genotype matrix. What are these centroids of the clusters themselves? T is we have a tree structure on the clusters. M, this is the model that captures the uncertainty in evolution. Alpha and beta is the model that captures uncertainty in the data itself. Alpha 0, we have to penalize complexity in the clustering. We don't want every cell to be in a cluster. What we use here is something known as the Chinese restaurant prior. It's actually a statistical model that is appropriate for clustering. Its job is to make sure that you don't just go crazy with the number of clusters. Because to fit the data well, put every cell in its cluster and it's perfect, right? And you don't want that. This is a parameter that governs it. So one point I want to make is that if someone looks at this, say, oh, these guys do very complex stuff. No, it's actually not very complex at all. If you understand what these are, which coming from the data collection process and from the evolutionary process, things make sense. The data comes from clusters. The data has genotypes that represent the clusters. There's a tree underlying them. We need to model uncertainty in the mutations. We need to model uncertainty in the data. That's all, right? But this is where understanding what's happening in the biology and in the sequencing technology lead to these kind of models. And I would say this is a simple model, but not any simpler. We couldn't do any simpler than this. We needed the error. We needed the uncertainty in the evolution. We needed everything here. Okay? We could have added more parameters. They wouldn't have helped us with more questions. The last thing I want to say here is that, where do these parameters that govern this, this alpha and beta error are governed by these things, and when you are doing in a Bayesian style, you can put prior on them from distributions that have to respect the data and what you are trying to model. Okay? Of course, computer scientists, where sometimes we differ from statisticians would have stopped at the previous slide. They say, here's the model, enjoy it. Computer scientists say we need to learn from that, right? I mean, we need to infer the model. That's not the model that someone gives us. We have the data, we want to infer it. So here, I'm not going to go about the details because this is not about uncertainty. It has uncertainty, as I will mention soon, but it has no issue with uncertainty in the data. Thus, the model is complex computationally now to infer. So we do something known as MCMC, Markov chain Monte Carlo to try to estimate the different components of that model, okay? So I'm not going to go into the details of this. But the one thing I want to mention that we talked about uncertainty in the input. There's uncertainty in the output as well. And the issue here is that when you do some modeling like this and you get inference from an MC Markov chain Monte Carlo does not give you a solution, say, here is the truth in biology. No, it actually gives you a big file that has 10,000 samples of truths. And you have to find something about them, right? And it doesn't give us a point estimate. It doesn't say here is the best model or here's the right model. It gives you a sample from what we call the posterior distribution. It's very important to summarize them so that you provide an answer to the biologist and put uncertainty in the output as well. Biologists never accept an answer that this is how the cells evolved. They would laugh at you. So there's no way to know. Actually, you cannot even verify that, right? So you can say this is how the cells evolved. I am 90% certain about this. Here's an alternative hypothesis. This has 8% confidence. Here's another one that has 2% confidence. So you give 3 or 4 and sometimes 1,000. Okay? So in our case, we use certain measures to do all these things and how to summarize and put uncertainty in the output. Again, I'm not going to go into the details. I will just finish by showing you what we get from all this once we did all the modeling. This is an example where we applied all this method to metastatic colorectal data from a cancer patient at MD Anderson. This is collected by our MD Anderson collaborators. This is the output from our method. We are visualizing it, of course. What this shows you is the tree structure that the method said is that there are cells. So the input was about 500 cells or so. It says that there was a group of normal cells. They are part of the input. These are healthy cells. At some point, these mutations started happening. So what you are seeing here, these are mutations in the genes. So these are names of genes. When these mutations happen, they give rise to this group here. And this group is in the liver now, okay? Because we know the organs there. Then there's another group of mutations that happened. Then they split into 2 groups, this group here, then this group and this group. So what you are seeing here is that what the method told the biologist is that you have 1 cluster, 2, 3, 4, 5. There are 5 clusters, and the heterogeneity in that patient has a level of 5. The cells are assigned to each one of them. The mutations that define each one of them are listed here. This is a subset of them. What's colored in purple here are genes that are implicated in cancers. These are tumor suppressors and so on. These are brakes on cancer. And these are mutations that were found by that. Once we get also this, we also have a hypothesis on metastasis. What our data shows, or the analysis by our method shows, is that the way that this cancer metastasized in this patient was through 2 paths. One here and one here. Okay, so these were actually, the metastasis did not go all in the liver. It actually went through 2 different paths of mutations. So this is very important information to the biologist because now they know the mutations in this patient, the order that they happened, the level of heterogeneity, and so on. Now they can start looking at this data, how the patient reacts to drugs, and try to start making some inferences about how this heterogeneity is playing a role, or which one of these mutations should be targeted for drug design. Okay? And with that, I want to just close with some thoughts that, as I showed you this, I was showing you this model that we inferred, and I was explaining it. In biology, explainability is very important. Just telling a biologist, here's a black box that says yes or no, they're not interested in that. They want something explainable. Simplicity is key to explainability. And we do need to define a model that's as simple as possible, but not simpler. Models are derived from observed data, the natural process that gives rise to the data, and the collection of data have uncertainty. They have to be accounted for. I am not a big believer of just let's throw math. It's, I have to take a dig on deep learning here. And I say layers of neurons. The solution is not to say we couldn't solve it with 1 billion neurons. Let's do 2 billion neurons and hope for the best. 2 billion neurons would not have done anything in this problem because we have a generative process and we need to uncover that. We do not need some box that has 2 billion neurons that, okay, they are saying it's a cat. It doesn't help us here, right? So understanding the biological processes and the technological artifacts are, to me, the only way to achieve these three. And with that, I want to acknowledge my former student, Hamim Zafar, who's actually also moving to our faculty position now, Ken Chen and Nick Naven at MD Anderson. Hamim contributed lots of these slides, and the National Science Foundation. Thank you for your attention. I'll be happy to take questions. I know it has nothing to do with what you guys do. I know that, yeah.
Really awesome talk. You mentioned the model couldn't be any simpler. What gives you confidence? Like, how did you know that you arrived at that?
Why we arrived at that, because all the components that we are putting in the model are what we need from it. So when we put the error model, when we put the mutational model and so on, these are components that we need in the model. If we eliminated any one of them, I could not have controlled anything about the error rate. If I removed the evolutionary process, I would not have controlled the rate with which C goes to T and so on. It's about the explainability. This is the fewest number of parameters that we needed to explain what's happening. We could have added more, right? I could have made one simple result in mathematics, the more complex models fit the data better. So you want to fit the data, just make the most complex model. We don't want that, right? I mean, we want to keep it as simple as possible, but we could not go below that because we didn't know how to incorporate error or mutational model or the number of clusters or anything like that. For example, one thing, I don't know what Derek is going to be what level of k-means Derek is going to be talking about, but in k-means, the k is the number of clusters, right? We could have said that parameter, the concentration in the Chinese restaurant process is not needed. We would have gone to a simpler model where we say, number of clusters is 5. But the biologist always will come back to the question, how did you know it was 5? So now you put that model and you hope for that the inference with the data will tell you that, right? So this is an example where we could have made it simpler, but it would have made it much more challenging to explain. Now I can explain to a biologist and say the number 5, the 5 clusters, that's actually what the data told us. It did not tell us 4, it did not tell us 6, right? So this is to me why I call it the simplest and not simpler than that.
So when you think about training the students, but when you're starting to turn to statistical methods, what are the core methods that you think practitioners need to know? What are the things that you think they should know coming out?
So the question is about, for the students who are being trained, computer scientists especially moving into statistical thing, because historically, I know why Derek is asking this question, because historically computer science students, statistics is not their favorite topic and they usually don't work with that. The field of computer science is becoming very statistical because of machine learning and these kind of things. I believe that fundamental understanding of probability is key. Fundamental understanding of probability, not just saying, I know how to write piece of code for doing MCMC. In fact, it's trivial, MC. You just propose a solution, evaluate, and see accept or reject. But why does it work? Why does MCMC work? What are these distributions? One of the things that when I moved into this field, I always was wondering when I heard the statistician talking about it. They will say, we assume Dirichlet prior on this and Chinese restaurant prior on that, and I always say, why? And they almost never would explain that. So it's important to understand these kind of probability distribution, these kind of processes, so that you know which one to apply. So for me, probability is really key for these things and having fundamental understanding of probability. I don't know if computer science students are reacting to that. I see at Rice so many students say, I want to do machine learning, but I don't want to do probability. And I say, that's an oxymoron. It's not easy to do these two, right? But understanding the basic distributions, basic characteristics of distributions like expected values, variance, how you compute these things, some basic theorems about these kind of things, I think they are very important.
Yes, so it seems like there was a certain level of heuristics being implied with your model. So you would talk to the biologists and get their input, and then that would kind of shape or this, in a lot of like machine learning type things, it's not really that, it's just like a black box, like you were saying. How important do you think these heuristics were to building what you ended up with at the end? Like these ideas from the biologists, like how much it was just, it seems like it was really high.
No, to me, this is crucial. I see actually I teach, the course I teach at Rice, it's called Algorithmic Thinking. I teach the student that when you want to do modeling in a discipline like social sciences or biology or whatever, I don't think the computer scientist should act as the biologist and the physicist and the social scientist, that we are the smartest people in the world and do all of that. The back and forth with the biologist is key. We have to talk to biologists, we have to understand the biology. So that, I don't know if, did I?
I was just trying to like gauge how much you thought came from the biologist versus what.
So how much came from a biologist? The biologist will tell us that there is error in the data. The biologist will tell us that infinite size assumption, I don't believe in it. The biologist will tell us heterogeneity is important in cancer. We don't know what the number of clusters and so on is. There's a tree structure underlying. And then the rest is us. Right? So what is the tree structure? What is the probabilistic model and so on? That comes from us. And going back to Derek's question, now we need to understand everything they are saying and say, well, this seems to fit an exponential distribution, or this seems to fit a Dirichlet prior or whatever. But I don't like to look at it as amount, right? Because maybe we talked to them for 5 hours and we worked for 50 hours. But in terms of the contribution, I would see it equal. If you want to have impact, it's really equal.
Does this work, when you show it to scientists, the biologists, change where they want to do future research, where before they were on one path and they look at this analysis and say, wow, if I change and go this direction, I can get a lot more out of that. Does that happen?
So it happens, so it's a very good question. I would say it again, is this recorded and going to be made public? So I will say that most people that I encounter in the medical field are very closed-minded. I would say it even if it's publicly available. They are resistant to trying things that are new. Now, the stuff we are doing is with people who are very open-minded. These are people who are doing this kind of experiment and this data. Is this going to change the mind of so many at this point? I would say that's a very big challenge. That's a very big challenge, right? So this is always an issue that when you are working in a new domain, even if you work like, I know Derek, for example, a lot of research with the, let's say, I don't know if we can call it social data, social informatic data. I'm sure there are social scientists that would never trust any computational analysis of data coming from Twitter or Facebook. They still prefer to interview people and so on. Same thing with biologists. They will still be resistant to some of these findings or relying on this new genomic data, and will say, well, we know how to do it and it's been working and so on. But I don't think one should give up, right? I mean, I think the onus is on us to show them what we get from this. And I think once you show them something that all of a sudden will tell them that this is really now impactful, they will change their mind, right? But if we, computer scientists, sometimes we make the mistakes also of developing methods and publishing papers. By, medical people and biomedical researchers saying, okay, publish. If they want to see it applied to data and how it works and so on. And the other key is that if you are doing it in a collaborative way with them, you listen to them and you incorporate that in the model, they are much more welcoming of the final result than if you work on it and say, I will tell you how biologists work. So, sometimes it's, the problem is with us, not with them. For some areas of biomedical research, actually, neural networks are used for image analysis. Biologists actually do a lot with image analysis, right? I mean, people, even at MD Anderson, they take CT scans and they want to compare before, after drug or CT scans of 50 patients. People are looking at that. Now, again, I mean, these are maybe funny anecdotes, but this is the reality. So we have a student at Rice who actually worked on this kind of image analysis, developing an algorithm to do image analysis. But when she developed the algorithm, she said, okay, how can I assess quality of the output, accuracy of my results? If I said this, based on this CT scan, this patient has cancer, I need some ground truth, right? So they took CT images, gave them, I believe, to 3 doctors or 5 doctors and say, now go each in your separate room and mark these ones. There was no agreement among them, right? So when there is no agreement among them and then you have a 4th black box saying something else, I don't know how you can actually validate these things. So at least with this, they are seeing something. You know, when we show them this kind of tree, we are saying, look, there is a mutation that happened in TP53, right? Black box, I think they will be even more resistant to that. Right? But I don't see, for example, how deep learning is applicable here. That's a separate question. I see it for image analysis and biomedical images, but for something like that, I don't know how it's applicable.
Yeah, so with this, I mean, so we're talking about uncertainty of outcomes, right? So is this a deterministic result of your...
No, I'm just showing here one summary from that results. Again, the results from our method, when I wrote 10,000, it's really 10,000. So you run this MCMC for like 10 million iterations, then you take samples from there, and there are statistical techniques for summarizing them, right? So we look at credible sets and so on. So no, in the paper, we do report these things, but here I'm just showing the results. Yeah.
Awesome, thank you.
Thank you very much.